So with that, I'll introduce our first speaker, Rui-Han Wei, a postdoc at the Johns Hopkins University. They'll be presenting their work looking at the ability to alter locomotor control in the behaving primate, titled Modulation of Deep Mesencephalic Nuclei to Locomotor Control in the Walking Monkey.
Good afternoon, everyone. I'm Rui-Han Wei from Johns Hopkins University. I'm in Colin's lab. Today my topic is modulation of deep mesencephalic nuclear to locomotor control in walking monkey. The deep mesencephalic nuclear is a large midbrain region. It is thought to play a key role in integrating information for sensorimotor processing like sensory, cognitive, and motor. Previous studies have shown that the electronic and the chemical stimulate of DpMe will elicit the posture changes and the locomotion, but the single neuron unit active to limb dynamics has not been characteristic. To modulate locomotion in response to self-motion, integration of vestibular information is required. DpMe cells also shows vestibular sensitive like rotation and translation, which is not moment related. And it remains unknown that whether if those same DpMe neurons would integrate vestibular and locomotion related signals. And also, how do these vestibular sensitive DpMe neurons modulate during locomotion without vestibular input is still not clear. And to address those questions, we did single unit recording of DpMe neurons. First, we applied passive translation and the rotation to characteristic the vestibular response of neurons in DpMe. And second, we recorded the same DpMe neurons during treadmill walking in absence of the vestibular cues. Four high-speed cameras were used for motion recording, and markerless motion capture DeepLabCut were used for extract the animal's posture. And for the result, first we identified 16 vestibular sensitive cells in DpMe. Most of them are sensitive to both rotation and translation. And here we show 3 examples from these 3 different categories. Like the first one is for the rotational cells. It is rotation. It is have high sensitive to the rotation stimulate, but during translation stimulate, it doesn't show modulate of this cell active. And next, we want to show that the DpMe neurons active during the head-fixed treadmill walking. For the first figure, what we showed is from the rest to treadmill walking, what does the kinematics and the neural actives like? And we show the ankle horizontal position and the knee joint angles during this paradigm, and also the corresponding firing rate. It is very clear to show that DpMe neurons are modulated with the gait cycle. And second, we compared the mean firing rate between rest and the treadmill walking, head-fixed treadmill walking, and it shows that DpMe neurons shows an increase during locomotion. And next, we want to show that the phase modulation during the DpMe neurons phase modulation during the head-fixed walking. And we presented the mean frequency distribution histogram of two types neurons during head-fixed walking. Also, the corresponding phase distribution of discharge frequency of neurons in 30 gait cycles. Modulation patterns varies between DpMe cells. Some cells demonstrate peak active during their stance duration, and some are peak active during a swing duration. Conclusion. We established that the same DpMe neurons that are sensitive to vestibular stimulation also modulate during head-fixed walking. During head-fixed walking, treadmill walking, DpMe neurons show sustained phase-dependent active changes and increase the mean variant. Furthermore, DpMe neurons show different in the phase dependence during head-fixed walking. And for DpMe neurons, it integrates vestibular and locomotion related signals. It is important for the generation of volunteer everyday activities such as walking. And our finding regarding the neural circuits that coordinate gait and balance have important implications for the development of the treatment and the interventions. And with that, I want to thank all the people in Colin's lab. Thanks for listening.
All right, that was really great. Super interesting. So you've got a few questions that are coming in over both the chat and the Q&A. I'm just going to remind people when they post to please ask whether you would, please also include whether or not you'd like to be asked to come on screen. I'm going to ask Omid Zobieri's question, which is, how did you extract the gait cycles? And then after that, we can begin bringing people on screen.
Okay. How I can extract the gait cycle? For the method, I use the camera to recording the walking for recording the video of during the monkey is walking, and then I use DeepLabCut. This is open source software. It could extract the markless motion capture. And for divided the gait cycle, I have the key point position from a monkey. So I extract like ankle position. So I just define the use left ankle angle to define the gait cycle.
Great.
So the start of the gait cycle is left ankle of the start of swing duration.
Great. So we have one question from somebody who's on stage. So Sean, why don't you ask your question?
Hello, everyone. Thank you for your nice work. And my question is the following. Since when you stimulate this region, you can initiate locomotion, then would you say that the neurons that you are recording now are like sensory neurons that receive information about locomotion, or do you think they are more like motor neurons that are actually controlling locomotion?
Yeah, for now we still don't know whether it is the for this DpMe neurons which responds during the head fixed walking. It is the motor information is input, or the DpMe neurons send the motor command to walking. We're still not clear with that.
Thank you.
I'll ask a question from Falanto. Have you studied the response of neurons to active vestibular stimulation? If not, would you speculate about the neuronal responses in that case?
Actually, we already started. We are studying the neural actives, the DpMe neurons active during the natural walking, like walking on the ground. It also have free. The head is free. We learned the active motion, active active vestibular input, and it shows like during when it walking on the ground, it will also increase the the active DpMe cells, the active is also increased and higher than the head fixed treadmill walking.
Great. And now we have Robin, who's on stage to ask her question. So you can unmute yourself and turn on your video if you'd like, Robin.
Okay. Hello, great presentation. I was wondering if you think that these neurons play an important role in maybe integrating different signals in order to modulate stance to swing transition, maybe as well as initiating locomotion, or maybe maintaining balance and like catching the center of mass with stepping.
Mm-hmm. Can you say it again? Sorry. Sorry, what is your question?
So given that the two example cells that you show have these different like stance and swing activity, do you think they play maybe some sort of role in modulating that stance to swing transition by integrating different sensory and motor signals?
Yeah. For this cells, DpMe vestibular sensitive cells. Some of them, I think, some of them have different functions, or they may have different sensitive for the inputs of the like perception from working, and also like the motor control from like some cells are related to the extender, some muscles, some are related to the flexor muscles. It all have these possibilities.
Great. And we have time for one final question from Farouche, who asks, what are the alternate motion capture methods? Why did you choose DeepLabCut over other methods?
For the monkey, we used we didn't use mark on the monkey because sometimes the monkey is like 4 limbs working. If we attach markers, the monkey will grab the markers, so it won't be stable. And also there are hair on the monkey, so if we attach the marks, it will like move, the marker will have movement, so it would be not that good. So we change it for the markless motion capture DeepLabCut. And for the DeepLabCut, we can have our own. We can build our own networks for training to capture the specific model of our own. So we choose DeepLabCut for analyze.
Great. So that is all the time we have for Rui-Han Wei. Thank you again for that great talk. We're going to move on to our second talk.